This lecture is going to cover macromolecules, but we'll go into some more advanced detail than what you already know about each of the macromolecules. So things that you'll learn about each one. We'll talk about what foods you can find each macromolecule in. We'll look at the structure in more detail than just identifying what the monomer of each is. And then there's some other information that's unique to each of the macromolecules that you'll find out as well. But be looking for food and structure for both of them. So we'll talk about carbohydrates first. We can find these in fruits. We'll find carbohydrates in vegetables, and also in any of your grains you'll find them. So recalling that our monomer is a monosaccharide, so taking a look here. So we're looking at glucose. That's our simple sugar that we're looking at in this situation. So when we bond multiple glucose molecules together, we get a polysaccharide, and the ones that we're going to focus on are starch, glycogen, and cellulose. So we're going to look at the structure of each of these, and you'll want to be able to identify which one is which and why each of them are unique to the things they're found in. So we're starting out with starches and glycogen. They are the most similar, and you'll see why. It's because of how those glucose molecules are bonded together. So we'll see pictures in a second. And then hydrogen bonds. With the hydrogen bonds in this situation, the hydrogen bonds are there to hold the molecules close together. So it's not the only way that the glucose molecules are attached, but it allows them to stay close to one another and not directly through a bond. So think how your water molecules get held closely together. Your glucose molecules are going to get held through water bonds, or through hydrogen bonds as well. So starches are branched chains of glucose molecules. So we have a bunch of glucose molecules that are going to be in a branched form. So if you look at this picture here, so each one of these right here is a glucose molecule, and then every so often you'll see one that branches off and moves in this direction. So this would continue, and then you'd see some branching. It would continue, you'd see some branching, is what we would see in this situation with the starch. So starches you're going to find in your plants, and one thing that can happen with starches as well is they can be broken down and used for energy as needed by the plant, because it's going to be broken down into glucose molecules, and glucose molecules are what is made in photosynthesis by plants. So the other one, also similar to the starch, is the glycogen, and you can see that it's similar in the way that it branches. So we have some branching this direction, we have some branching this direction. So that's where those two are similar. It is a little bit more advanced in the branching than it is in plants, and you will find glycogen in animals. Then our last polysaccharide that we're going to look at is cellulose. So very different in structure in terms of that it is very straight. So you can see each one of these is still a glucose molecule, but they are bonded in a straight line. So cellulose we are going to find in plants, and it's one of the major building blocks in providing plants with their strong and sturdy structure. It's going to be found in the cell walls, and it's very tough. So if you've ever bit into some sort of plant, so let's say celery is a good example, that tough stringy part that you eat when you're eating a piece of celery, that's a lot of cellulose. So it's very strong and it's very rigid. All right, next we have lipids. So just recalling that our lipids do make up cell membranes, but your lipids can also be broken down as a usable piece of energy as needed. So if the carbohydrates are not there for an energy supply and extra energy is needed, we can use the lipids for that. So in terms of where lipids are found in animals, we'll find them in meat and things like butter, milk. So those lipids that are found in animals are mostly going to be saturated fats. So if you remember, your saturated fats are the ones that do not have any double bonds. We're looking at all single bonds between those carbon molecules. And then in plants, you would find it in things like olive oil, peanut oil, any of your oils that are plant-based. And these are mostly unsaturated fats. So remember that these are going to have the double bond in them. Now, it is because of this double bond that the molecules are not as closely compacted together, because what that double bond does is it makes like a kink in the, so we have a carbon, makes a kink in the chain, which makes it so two different chains of fats cannot bond so closely together. So if that's one, if I had another one that was here, there's going to be some space between them. So this space right here, because of this double bond, they can't get as close together as opposed to one that would be your saturated, where they could be right next to each other. So because of this space, your unsaturated fats are the ones that are mostly in a liquid form, as you can kind of see when you're looking right here. Your plants are your liquids, your animals are more things like solid fats that you would find in meats and butters. So a phospholipid is just a specialized type of lipid. So when we are looking at the structure on this, it's very similar to the triglyceride, but what it has is the addition of a phosphate molecule, which you will see right here. So that's a phosphorus with 4 oxygens around it. Oops, sorry. So we still have the fatty acid tails. In a phospholipid, you're only going to have 2, but you still have the tails. So here's your tails. They are hydrophobic. So think if someone has a phobia, they are afraid of things. So the tails are afraid of water, and they are nonpolar, which makes sense because we know that polar things dissolve other polar things. So these tails do not like water. They want no water. So then the phosphate and the glycerol, when we combine them together, we say that that's the head. So that's right here. And it is hydrophilic, meaning it loves water, and the head is polar. So it wants to be close to water. So we like water up here, and we do not like water down here. Another type of lipid that we have in living things are cholesterols. So cholesterols are shaped very similar to our traditional lipids. So if you think of when you built the molecule, what the lipid looked like, except instead of just being a straight chain, now we have rings that make up that line. So each of these rings is a carbon ring. So it's a bunch of carbons that are connected to each other. Sorry. So cholesterols are also found in the cell membrane. They're used for transmitting signals, and we'll talk more about that a little bit later. But they also make steroid hormones. So these hormones could be used for regulating the response to stress in the body, and then they're also used for sexual development. So things like estrogen and testosterone are examples of cholesterols that could be found in the body. Next, we have proteins. So with proteins, remember that the amino acid is the monomer, there are 20 amino acids that living things are going to use to make proteins. So they are all different, they are all unique, and there are 20 of them. However, our bodies themselves can only make 12 of them, so the other 8 must be consumed for the body to be able to make them. So when you hear about people, oh, you need to have protein in your diet, you need to have protein in your diet. Well, it's because your body needs those amino acids that are provided by those foods. So where can we get those? Meats will provide those amino acids, things like beans, nuts. So anything you generally hear, oh, that's high in protein, it's because it contains the amino acids that we need to make the different proteins in the body. So just a reminder that all amino acids do have a similar structure. So this picture right here should look familiar to you. So we have our amino group on this left side here, our carboxyl on the right. Everything's going to have hydrogen, and everything is also going to have an R group. So now let's look about what each of those means. So the things that you're going to see the same on every amino acid is, let's change colors here, the 3 here. Your amino group, your hydrogen, and your carboxyl, all going to be the same. So it's this R group right here that is going to be different. That R group is going to be what makes each one unique. So it depends, whatever is on attached in this area right here where the R is, that's going to be what makes each unique amino acid. So we know that when we join amino acids together, it's going to eventually give us a protein. But what are those amino acids joined by? They're joined by covalent bonds. So it's a covalent bond that is going to join chains of amino acids together. So I have my first amino acid joined by a covalent bond to my second amino acid. And what those specifically, the covalent bonds in a protein is called is a peptide bond. So peptide bonds are specific types of covalent bonds. So they're going to form between the amino group of one to the carboxyl group of another. So if you look back at this picture right here, next to this side by my carboxyl, I join 2 amino here. On this side there would be a carboxyl that would be joining to this amino group right here. So when I link amino acids together into a chain, they're called polypeptides. So remember, poly, many. Here's our peptide. So that's what the bond specifically is called between the amino acids. So I have many peptide bonds. And then when I join a bunch of polypeptides together, actually one or more, that's when we actually get a protein. So this picture down here is going to show you a peptide bond. So you can see the carboxyl here joining with the amino here to give you, this is now your bond. So that's your peptide bond. And one thing that you will notice, and we won't go into a lot of detail about this, is this OH and this H go away. Well, what's OH and H give me? Water. So every time I form a peptide bond, water is released as a waste product. So one thing to keep in mind about proteins is every protein is unique depending on what its function is going to be in the cell. So if you think about all of the functions that proteins have, it's a lot of different things inside of living things. So they're all unique. They all have different jobs, so they're going to be different. And now, how are they going to differ is depending on the order of the amino acids and the number of them. So there's 20 different ones that we have, so there's a lot of different combinations that we could have when making them. So it's the specific sequence that will determine the structure and the function of the protein. So when we're talking about structure of protein, there are 3 levels that we'll talk about. The primary level of protein structure is what we call it. It's just the sequence of the amino acids. In what order are they? So as we look at DNA and we start sequencing proteins later in the year, we'll be writing out the primary structure of each protein that we're looking at. The secondary level of protein structure is the arrangement of space, so what it looks like physically and how it's going to bond. So there's 2 main shapes that you're going to see, and they are listed for you over here. So my suggestion would be to kind of sketch out what these look like. It doesn't have to be perfect, but just so you have an idea here. So the first is the alpha helix. So if you remember when you first learned about DNA, you learned that it was a double helix. So anything helix means that it is twisted. So the alpha helix in this, our first structure, is our protein is twisted, kind of like a staircase, a spiral staircase. And why it is like that is there are hydrogen bonds that form between every 4th amino acid. So if you look here, these yellow dash lines that I'm now making purple are showing the hydrogen bonds. So these are bonding between the amino group of one amino acid, so the H of one amino group, to that double bonded oxygen on a carboxyl of another. So that H and that O are going to be attracted to each other, and what's going to happen is they're going to start to twist because of the shape. So it kind of twists upon itself like a ladder. So that's your alpha helix shape. The other that we have is called a beta sheet. So this is when we're going to have some parallel lines of amino acids. So you can see we have one right here, and we have another line of amino acids here. And our hydrogen bonds are just going to form parallel between those 2. So once again, they're yellow here, but I'll highlight them in purple. So you can see we're going to have hydrogen bonds that are forming here. So this allows for these to stay close together. Now, there's nothing special about each one of these. It just is what the function of the protein is and what shape it actually needs to be. And then the tertiary is the overall shape of the protein after it starts to fold and what it eventually becomes. So this tertiary right here, this is the final shape of the protein. So last but not least, we have nucleic acids. So one thing to keep in mind is nucleic acids are not found in foods that we eat. So we will not be finding them in there, but you will find your carbs, your lipids, and your proteins. So let's talk a little bit more about our 4 bases. So we have adenine, thymine, cytosine, and guanine. So their structure on them is a little bit unique for each one. They either are a single ring or they are a double ring, and I will put a picture up for you on Classroom of what those rings look like. But you want to know the names of the bases, and you want to know if it's a double ring or a single ring structure. Because when we talk more about the DNA structure, when we do DNA a little bit later, that double ring, single ring part is going to be important. So the next thing is that you're going to have covalent bonds that are going to form. And the covalent bonds, so if you think about the structure of a single nucleotide, so you have your 5-carbon sugar, you have your base, and you have your phosphate. Let's say my base is here. This part right here that's going to bond together that I just highlighted in yellow, that's going to be covalent bonds that are going to form to actually hold the nucleotide itself together. And then what happens is we're going to have hydrogen bonds that are going to form between the bases on each side. So that's what's given to you here. It's highlighted. So think of DNA as being a ladder. So it's going to hold that ladder together in the middle is going to be hydrogen bonds that are going to attract one side of the molecule to the other. So in each of these spots where your colors are not the same but they are connected is hydrogen bonds going all the way down. So even though a hydrogen bond on its own is not very strong, when I add them all together and it is there holding the entire strain of DNA together, it does become very strong and it's able to stick together as one. So that is the end of this video. If you have any questions, please let me know.